Hey guys, welcome to the urinary system. Today we're going to describe the functions of the urinary system in terms of effect on blood pressure, blood chemistry, the endocrine function, as well as waste removal. We're also going to talk about the structures of the urinary system and then talk about the physiology and stages of urine formation. Oops. So let's talk about the functions first. So the functions are excretion, elimination, water balance, blood chemistry, and the production of EPO, which helps with blood pressure regulation, but also specifically with hematopoiesis, which is the production of red blood cells. So for excretion, we're going to remove any waste material. This removes the waste material from our blood plasma, and then we dispose of that waste within the urine. We also do elimination. So again, removal of the waste from our organ systems, from our digestive system. We're going to get rid of any undigested food, water, salt, ions, and drugs. From our respiratory system, we can get rid of carbon dioxide, hydrogen ions, water, and toxins. From our skin, we can get rid of water, sodium chloride, nitrogenous waste, including urea, uric acid, ammonia, and creatinine. We're going to do water balance. So this is where our kidney tubules are going to regulate our water and our urine concentration and our blood chemistry. This is how we regulate our pH, volume, and composition of our body fluids. And again, the production of EPO to help with hematopoiesis and renin for blood pressure regulation. So for a urine, our endpoint is going to be this urinary system urine, right? That's our endpoint. And we want to make sure that it consists of water, electrolytes, and waste products. Urine should not contain any white blood cells. Any white blood cells in our urine indicates that we have an infection. Glucose or ketones can indicate diabetes, and blood or protein can indicate any renal damage, like a kidney stone potentially, or even infection. Our urine is going to vary from clear all the way to a dark brown, just sort of depending on sort of our hydration level. So if we're super hydrated, we're here in this region, and if we're dehydrated, we can be all the way in here, this region. Our output is going to range from about 3/4 to 2 liters per day with good hydration, and our daily water intake should be about 9 cups of fluid for females and about 11 cups in males to maintain hydration. If you're in a hot climate, or you're doing exercise or a lot of work outside, or if you're sweating a lot, then you're going to need more. The darker the urine and the higher the specific gravity of the urine, then the more dehydration. So let's talk about our urethra first. Okay, and so here, this is our female. So here's the urinary bladder, and then that's the urethra heading out towards the labias. And then here is the male urinary bladder, and then there's the urethra going through the penis. So our bladders, here's an example of a bladder, and the urine, what happens is that it comes down through the ureter into the bladder. And the bladder has different layers. So we have the peritoneum. Remember that the peritoneum is that covering over our tummy organs. So our abdominal cavity, right, is covered over in the peritoneum. And so the bladder is outside of the peritoneum. Here are the inside of the bladder. This is what's called the detrusor muscle, which is going to sort of surround that urinary bladder. And here is a zoomed in vision of that. So you can see that this is transitional epithelium. There's that lamina propria, and then this is submucosa. Okay. And then we have these ureteral openings coming in, and then down here we have what's called the internal urethral sphincter, because these are ureters. And then this is your urethra, okay? So we also have some nerves that are going to help us to identify that our bladder is full. So we have what's called the pudendal nerve along the back. And then our kidneys here are a pair of bean-shaped organs, and they're located retroperitoneal. So they're going to be on the outside of that peritoneum as well. They're on the back surface of it, in fact. And so that's, if we have any kidney infections, they're not going to be able to spread to the abdomen. The kidneys are responsible for our blood filtering and the urine formation. So you can see there's the kidneys, and then the urine would travel down through the ureter to be stored in the urinary bladder, and then we would go down into the urethra to be excreted. Okay, so our kidneys have different layers. So the outer cortex along this is where we have what's called the glomerulus, and this is where our blood gets filtered. And then we have different types of arteries out here. So you can see here, this is our renal artery coming in. The blood's going to come in and it's going to go through these interlobers, across the arcuates, and then into these cortical blood vessels. And then we're going to get sort of that transfer and then back into the renals. Okay. So we have these radiate arteries out here. They're called cortical radiate arteries. And we have afferent and efferent arterioles, and we have the paratubular capillaries in there. We also have the proximal and distal tubules for reabsorption. The inner medulla, which is this area, are going to contain the renal pyramids. This is the renal pyramid right here. Okay. And in there, we have what's called the loop of Henle for water balance, collecting ducts to collect the urine, and we have some major and minor calyces. And we're going to talk about those a little bit more, but there's a minor calyx and a major calyx here. And then we have the hilum. This is the renal hilum, which is an area. That's where the area where we have the renal veins and arteries and the renal nerves coming in. And also within here, we have what's called the renal pelvis, and the renal pelvis is going to direct the urine into the ureter. Here's that ureter. Okay. So here's our blood flow. So what happens is that we get the blood coming in through the renal artery. Then it's going to branch into what's called a segmental artery. Then we have our interlobers, an arcuate that is going to go across the arch. And then it's going to go into our interlobar arteries here, okay? And so there's that interlobers, and here we're going to get a little smaller. So we have what's called an afferent arteriole. Afferent means that it's arriving, and so it's arriving to this area here, which is called the glomerulus, okay? And then we have an efferent arteriole here, okay? And so that afferent is going to bring it in, and it's going to pass through this glomerulus, and then it's going to come back out. That's where that efferent. And then we have what's called the paratubular capillaries out here, and that's because they're going within this tube. And then we're going to come back in through our interlobar vein. Here they are there, arcuate veins, and then into our renal vein there, and then coming back out. This is our nephron, and the nephron is considered our functional unit, and it's composed of 10 parts. So we have this afferent arteriole coming in, we have the efferent arteriole, we also have this glomerulus. This is the glomerulus capsule is what this is called. We have the proximal convoluted tubule, proximal because it's the closest to the glomerular, and then convoluted because it's kind of messy and it's a tube. And then we have going down through our loop of Henle, right there. And so going down, we have a descending limb and then an ascending limb of our loop of Henle. Then we have our distal convoluted tubule, and then we're going to go into our collecting duct, and it's going to travel into that renal papilla. So we look at our microscopic images. This is a picture from a microscope to show this glomerulus, and you can see that afferent and the efferent arterioles coming out. This is called BOWMAN'S capsule right there. So the way that urine actually forms is that we have filtration, reabsorption, secretion, and excretion. One of our steps, though, with that sort of idea is that we also have concentration. So filtration is happening right here at the BOWMAN'S capsule. So these small molecules get filtered from our glomerulus capsule. So remember, there's blood in here, right? And so that blood is going to be filtered out, and we go into the BOWMAN'S capsule. Then we have some reabsorption happening. And so reabsorption is happening along the proximal convoluted tubule and the distal convoluted tubule. And they do that at the paratubular capillaries. So there's the paratubular capillaries here. Okay. And then we have concentration. And so concentration is where this water gets reabsorbed from our descending limb of our loop of Henle, and then from the collecting duct into the paratubular capillaries. And then we have secretion. And so secretion is where we're going to get rid of any waste or harmful substances. They get transported from our paratubular capillaries to our proximal convoluted tubules and our distal convoluted tubules. And then eventually we're going to excrete them out. So our BOWMAN'S capsule, or our glomerulus capsule, has different layers. So we have the visceral layer and the parietal layer. So the visceral is going to be right on the organ, and then the parietal is going to be off of the organ, right? So the visceral layer has what's called podocytes. And podocyte actually means little feet with these pedicles on them. And they have these sort of modified simple squamous cells that are lined along this visceral layer of our Bowman's capsule. Each cell has these several primary processes, and each primary process gives rise to these secondary processes that we call pedicles. Okay, they're going to end with these sort of feet placed on the basement membrane of this glomerular capillaries, and it creates this filtration membrane, almost like these. We have like these filtration slits sort of all along this capillary tube. And the capillary is a fenestrated glomerular capillary. So remember, fenestrated capillaries are going to have all of these sort of holes in them. And then the parietal layer is a simple squamous epithelium. So the plasma of our blood gets filtered by the filtration membrane, and it has those fenestrated capillaries of the glomerulus and those podocytes with the foot processes. And the fenestrations and the foot processes are going to limit the size of what can be filtered because the blood cells and the proteins are too large to filter through. So we have to see what's going to actually filter. So we have water, glucose, amino acids, peptides, electrolytes like sodium, potassium, chloride, bicarbonate, calcium, phosphates, vitamins, minerals, and nitrogenous waste. Nitrogenous waste is urea, uric acid, and creatinine. Creatinine is, all of these come from this breakdown of protein, DNA, and creatine. So what happens is that we have all of these hydrostatic pressures that you may remember that we talked about hydrostatic pressure and osmotic pressure before, okay? And so all of those pressures are actually going to be filtered at that glomerulus. So we have our blood hydrostatic pressure. Remember, hydrostatic pressure is that push out, okay? And here at the glomerulus, the pressure is about 55 millimeters of mercury. Then we have the capsular hydrostatic pressure. The capsular hydrostatic pressure is about 15 milligrams of mercury. Now, the capsule surrounds the blood sort of vessels, right? And so it's a push in here. And then we have our blood osmotic pressure. So we talked about that blood osmotic pressure and how with the blood osmotic pressure, we're getting the pull back in, right? And we have that because of the plasma proteins within the blood itself. Okay, and so we have this net filtration pressure. So we have our blood hydrostatic pressure minus the capsular hydrostatic pressure and the blood osmotic pressure. Okay, so if we have our 55 minus 15 plus 30, we get 10 millimeters of mercury. And so this is a net push out from our blood, which is going to give us filtration. Okay. So if we see here, we have this blood hydrostatic pressure. So this is our blood right here, right, coming through these glomerular capillaries, okay? And they're pushing it into this capsule right here, okay? And then we have this blood colloid osmotic pressure, right? It's pulling it back in. But we also have, because there's fluid in here, a hydrostatic pressure against this tube. But that hydrostatic pressure is not as strong, and so it's pushing back this way. So we get about 10 millimeters of mercury of this net outward pressure pushing into this capsule, into the Bowman's capsule. So we get what's called a glomerular filtration rate, and we estimate this most of the time. So this is the volume of filtrate that can be formed by both kidneys per minute. Okay, so if our cardiac output is about 5 liters per minute and 20% of that, or 1 liter per minute, perfuses into the glomerulus. On average, this is going to result in the production of about 125 milliliters per minute of filtrate in men and about 105 milliliters per minute of filtrate for women. So 90% of the filtrate gets returned back to our blood volume. So then about 1 to 2 liters of urine gets produced daily. So we have what's called the JGA apparatus. This is called the juxtaglomerular apparatus. Okay, and so here are the juxtaglomerular cells right here. This is called the macula densa. There's the JG cells there, okay, and so we have our afferent arteriole, the blood's coming in, right, here are these, the podocytes are all pressing onto that, and then this is that efferent, right, and so we're bringing in, okay, and then the fluid would be going into this capsule right here, but these are called the JG cells, or the juxtaglomerular cells, okay, so the JGA is going to sort of tell us whether we're dehydrated, right? Because when we're dehydrated, we're going to have less of a pressure coming in, right? And so we're not going to have sort of that same push against what are called the macula densa cells. So those are the macula densas. Okay, so if the osmolarity of our filtrate is too high, as noticed by those macula densa cells, because we would have dehydration, which is going to cause low blood pressure, those macula densa cells are osmoreceptors. Osmoreceptors are then going to trigger those JG cells to release paracrine hormone adenosine, which is then going to trigger our granular cells to contract and decrease the blood flow to the glomerulus, which is going to decrease our glomerular filtration rate. Okay, so in times of low blood pressure, so low blood flow, we're going to have to decrease our blood flow to the glomerulus. Okay, then we're going to have less plasma filtered, leading to less urine formation and a greater reabsorption of water. Okay, and so hopefully that's going to ultimately decrease our blood osmolarity, correct our dehydration, and maintain our blood pressure, right? So let's just review that one more time. When we have low blood pressure, like when we are dehydrated, okay, when we're dehydrated, we're going to have more osmolarity in our blood. So our blood is like saltier, right? So those macula densa cells, which again, sorry, are right here. Those are the macula densa cells in the green. They're going to sense the osmolarity, okay? Then they're going to cause the JG cells to release a hormone, adenosine. Adenosine is going to then trigger those glandular cells to contract and decrease the blood flow to the glomerulus, which is going to decrease our glomerular filtration rate. Then less plasma gets filtered, less urine gets formed, and we get a greater reabsorption of water. Hopefully, that's going to decrease our blood osmolarity because we're putting that fluid, we're keeping it into our blood, correct our dehydration, and maintain our blood pressure. Okay, so we have this whole mechanism, which we've talked about this before, but let's go over it again. This is the renin-angiotensin mechanism. Okay, we talked about it as the RAA pathway. Okay, so there's those macula densa cells. They sense that low fluid flow or a low sodium concentration. Okay, and so then we're going to have those JG cells secreting renin. Okay. So the liver is always releasing this angiotensinogen into the blood, okay? But when we get that renin in there, it's going to cause us to have that enzyme reaction, and we're going to convert to ANG1 or angiotensin 1. Okay. Angiotensin converting enzyme is within our lungs, and so we're going to get another enzyme reaction, and so we're going to convert to ANG2. ANG2 is going to cause us to have widespread vasoconstriction. But we're also going to stimulate our adrenal cortex to secrete aldosterone. Remember, aldosterone is going to stimulate our sodium uptake on our apical cell membrane into the distal convoluted tubule and collecting ducts. And this is new, we're going to have ADH. ADH is antidiuretic hormone. An antidiuretic hormone is going to cause aquaporins to move to the collecting duct. This is the collecting duct right here. Okay, and that's going to increase water reabsorption. So if we're reabsorbing the water, remember the water is going to follow the salt, so that's what that is. Okay, and so what happens is whatever stays in this yellow tube is going to leave our body. That's going to become urine. Okay, and so we have to reabsorb the water. If we reabsorb the water here, our urine gets more concentrated and darker because we're dehydrated, right? And the water gets reabsorbed back into our body. Okay. So this is how our tubular reabsorption and secretion happens. So we have the proximal convoluted tubule. So there's the glomerulus. This is the proximal convoluted tubule. So at this point, we get reabsorption of about 65 to even up to 100% of the solutes. So we reabsorb it back into our body. Glucose, amino acids, proteins, vitamins, lactate, urea, uric acid, sodium, potassium, calcium, magnesium, chloride, bicarbonate, and water. All of it gets reabsorbed back in. And we also, at this point, are going to excrete urea, uric acid, creatinine, and some drugs. They're going to go into this tube so that they can eventually become urine. Okay. Now, then we get to our loop of Henle. And at our loop of Henle, we're going to reabsorb water. So water's coming out of the tube. Okay. at our distal convoluted tubule, which is over here, we get the reabsorption of calcium. Okay, we get reabsorption sort of sodium, chloride, bicarbonate, and more water. Okay, and we're going to get excretion of hydrogen ions, potassium, and nitrogen. Then our collecting duct, which is right here, okay, we're going to excrete any acid and bases, and we're going to reabsorb some water. So this proximal convoluted tubule, so all this filtered fluid collected by our Bowman's capsule enters into that proximal convoluted tubule. It's called convoluted because it's just like a mess, right? And then there are simple cuboidal cells form the tubule with this prominent microvilli on the luminal surface, and they form a brush border. The brush border is very similar to that brush border that we talked about when we talked about the small intestine. Many of those mitochondria are going to run the active transport pumps that are used for the reabsorption of solutes. We also get some facilitated diffusion here. And that immediate reabsorption of about 80% of the solutes in filtrate, glucose, amino acids, and those electrolytes, vitamins, and minerals, and the secretion of nitrogenous waste. So again, proximal convoluted tubule, immediate reabsorption of all those solutes, glucose, amino acids, electrolytes, vitamins, and minerals, and we're going to secrete out urea, uric acid, creatinine, hydrogen ions, and ammonium ions. Okay. Then in our proximal convoluted tubule, as we continue, okay, just sort of to kind of go over it, glucose, amino acid, peptides, vitamins, minerals, filtered water, sodium, potassium, calcium, and the bicarbonate and phosphate groups, urea, uric acid, creatinine, ammonia, and hydrogen ions are secreted out, right? So they're going to go into the tube, and these are going to be reabsorbed out of the tube, okay? Then we have the loop of Henle. Okay, and so we've moved from the proximal convoluted tubule down this loop of Henle. Okay, and so these juxtamedullary nephrons are going to extend into the medulla, which is the center area of the kidney, and they have all of these vasorecta capillaries for water reabsorption. We have some thin portions of the loop of Henle. Those thin portions, like the descending loop, have simple squamous epithelium, and they have these permanent aquaporin channels. And aquaporin is just a way for water to go through. We also have these receptors for ADH, which is antidiuretic hormone. So when stimulate, those cells can insert more aquaporins to reabsorb more water when we're dehydrated. Okay, so we can do that RAA pathway. And then we have some thick portions. The thick portions are this simple cuboidal epithelium. That's the ascending loop that's going back in, and they're going to create this medullary concentration, which is called a countercurrent mechanism or countercurrent multiplier. So this effect on this vasorecta capillaries is that we can get more reabsorption of sodium, potassium, and chloride. So those ions are going to help to move the water. Okay, where sodium goes, the water is going to follow. That is key. Okay, and so what happens is that we do this multiplier, and so we sort of pump out. This is active transport here, meaning that we're using energy, and so we're pumping out the sodium and chloride. Okay, and so when we do that, when we pump it out, that means that we're going to then get this water diffusing through. Okay, and so this is passive transport, meaning that it's happening through diffusion. No energy is required for that to happen. Okay, then, so you can see here this sort of numbers of concentration. So here down here, this urine is highly concentrated here. Okay, and one of the reasons that it's so concentrated is because, look, so we pumped out the water. The water is diffused out, right? And so it's diffused out, no energy. Okay, and it's done that because of this sodium chloride up here. Okay, but we've also got some diffusion of sodium chloride here, and we're putting in some urea. Okay, and so by the end of the time, what happens is that we have about 100 milliosmoles here of concentration. Okay, and so that's the concentration of the fluid within the urine, and so that's pretty typical for urine. That's what it sort of should be if you are hydrated well enough. Okay, but what happens is if we're dehydrated is that we're going to put in those ADH aquaporins. We're going to get even more water out of here, and so it's going to be even more concentrated, and then we're not going to be able to fix it before it gets released, meaning that this is going to be highly concentrated. So remember, anything that goes into this space is going to end up back into the blood. So there's the blood right there. And so you can see this blood of our efferent arteriole. Okay, it gets concentrated. Here's our vasa recta. It gets highly concentrated. But then we get some passive transport movement, and it's not going to be as concentrated when it gets to the veins. Okay. So that distal convoluted tubule is made out of simple cuboidal epithelium. It's a pretty rare microvilli. And this is where we get some hormone-mediated reabsorption. So PTH, parathyroid hormone, is going to trigger calcium reabsorption when our blood calcium levels are low. PGH is also going to trigger the activation of 1,2,5-OH vitamin D3 to increase our calcium reabsorption from food in the intestines. Aldosterone is going to trigger the reabsorption of sodium, and water follows. ADH triggers the reabsorption of water, and ANP, atrial natriuretic peptide, is going to trigger sodium excretion, and the water is going to follow. So the reabsorption of about 10 to 15% of our filtered water by our ADH and aldosterone can happen during times of dehydration. We also have these collecting ducts. The collecting ducts are the last chance for us to modify the filtrate of what's going to stay in the tube and what's going to come back into the body. So we have what are called principal cells, and these are triggered by ADH and aldosterone to reabsorb that remaining water, and virtually 100% can be reabsorbed during times of dehydration. When stimulated by ADH, these cells can insert additional aquaporin channels to reabsorb water during dehydration. We have intercalated cells that are involved in our acid-base homeostasis, and so they can dispose of the acid generated by our dietary intake. Hydrogen ions get excreted in exchange for potassium ions, and the remaining filtrate at the end of the collecting duct is going to become urine. So this filtrate enters this sort of convoluted tubule, okay, and it's isomotic to both the blood plasma and the cortical interstitial fluid. So this is the cortex of the kidney, okay, so the inside. This is the medulla, and then so the outer medulla, and then the inner medulla, okay. So this is the outside of the kidney, middle, and then very middle, okay. And so here you can see that they're pretty similar in their osmolarity, okay. And then what's going to happen is that the water is going to move out of the filtrate in the descending limb, and it's going to move down as the osmotic gradient, and that's going to concentrate the filtrate inside the loop. The filtrate is going to reach its highest at the bend in the loop, and then the sodium chloride are going to be pumped out of the filtrate. That's going to increase this interstitial fluid osmolarity, okay? And then filtrate is at its most dilute as it leaves the nephron loop at about 100 milliosmoles. It's hypoosmotic to the interstitial fluid. Okay, so here we can see this formation of the dilute and the concentrated urine. Okay, and so when we don't have those aquaporins in that ADH, okay, then we're going to just get our normal urine creation. Okay, but when we have aquaporins inserted, they're going to be inserted here. So we're going to have water is going to start moving, see? And so because once it gets into this tube, the water's not moving anymore. Only the urea is, and it's moving through passive transport. But the water no longer is, right? So up here, okay, when we have this presence of ADH because we're dehydrated, we're going to move that water out because we put in these aquaporins, so the channels for the water to move, and so that's going to highly concentrate the urine. Okay, so let's review. Let's talk about the functions of the urinary system in terms of its effect on blood pressure, blood chemistry, endocrine function, as well as waste removal. Describe the structures of our urinary system, and then the kidney's physiology and the 3 stages of urine formation. Okay, so here are our sort of functions, right? And so we're going to excrete, remove that waste material from our blood plasma. We're going to eliminate any waste from other organ systems. So our digestive system is going to get rid of that undigested food, water, salt, ions, drugs. Respiratory system can get rid of carbon dioxide and hydrogen ions, right? We're going to do water balance here. So they're going to regulate that water reassertion and the urine concentration. We're going to do blood chemistry. So we're going to regulate the pH, the volume, and the composition of our body fluids. And we can produce EPO for hematopoiesis, okay, and the renin for blood pressure regulation. Okay, so as far as blood pressure, remember when blood pressure is low, we're going to activate that RAA pathway. And so we have the renin for blood pressure regulation, but we also have that ability to insert those aquaporins, okay, and concentrate the urine even further to hopefully bring back our blood pressure numbers, okay. So when we look at our sort of what's happening, okay, so this is the cortex, this is the medulla right here, okay, and so this is our outer layer of our kidney, and then in towards the center, and then the very middle, okay. So we have this glomerulus, okay, we have the afferent tubule, the efferent tubule, there's the glomerulus, the Bowman's capsule, and so what happens is that we have such a pressure, right, that we're going to get this push over here because the blood hydrostatic pressure is stronger than the osmolarity, the blood osmotic pressure, as well as the Bowman osmotic or the Bowman hydrostatic pressure, right? And so that's going to get this push out, okay? And so we're going to do the filtration. Now, once we get out here, and then this proximal convoluted tubule, we get that immediate reabsorption, we get the glucose, the vitamins, the amino acids. And we're going to secrete out the urea and the ammonia and all of that good stuff, right? And then, okay, we're going to start to see that movement of water, okay? And we're going to highly concentrate what's in the tube. One of the ways that we highly concentrate that is by the movement of urea. Then we're going to start to see the sodium and the chloride move out, okay? As we move up through the distal convoluted tubule, we're going to start moving, see that water move out even more, right? And so the osmolarity of this is going to decrease to about 100 milliosmoles. However, if we are dehydrated, we're going to see that insertion of these aquaporins in here because of ADH, and that's going to cause the water to move back out into the interstitial fluid here, okay? And it's going to go into the vasa recta because there are blood vessels all throughout here, right? All those blood vessels. So it's going to reabsorb back into the blood and hopefully bring our blood pressure back to normal. Okay. So here are some hormones as we see our activation. So what happens is that atrial natriuretic peptide is going to decrease this glomerular filtration rate, whereas renin is going to increase this glomerular filtration rate. Okay. Now, once we can do that, we also have the ANG2, which is going to activate here. So it's going to increase that sodium and water. We have the aldosterone here. There's some more ANG2, and then the ADH is going to insert in those aquaporins. So this is where our blood pressure gets sort of messed with, I guess, okay, with our hormones. So what I want you guys to do is take a look at this blood pressure, our hormonal effects, and see if you can fill in this chart. So ADH, aldosterone, ANG2, renin, and atrial natriuretic peptide. So first, where is it produced? When does it get released? What does it do to the nephron? What's it going to do to our overall body response, our blood volume, and our blood pressure? So pause here and see if you can do that before you move on. So when we can see our ADH, which is also known as vasopressin, okay, antidiuretic hormone, it's going to be produced in the hypothalamus and then stored in the posterior pituitary. It's going to be released when we have dehydration, and it's triggered by the release of ANG2. Okay, now that is then going to cause this aquaporins. Well, it's not triggered by the release of ANG2, but you know what I mean, triggered by ANG2. The aquaporins are going to be inserted in our convoluted, our collecting duct, so the water gets reabsorbed. When we have less water loss and a vasoconstriction, that's hopefully going to increase our blood volume, which is then going to increase our blood pressure, okay? So ADH is going to increase our blood pressure by increasing our blood volume. Aldosterone gets released or produced by the adrenal glands, and it's going to be released when we have low sodium, which is hyponatremia, low blood pressure, hyperkalemia, which is an increase in potassium, and it's also triggered by ANG2. Okay. The distal convoluted tubule is going to reabsorb more sodium and water. Less water and sodium loss is our overall goal here. And hopefully we're going to increase our blood volume and therefore increase our blood pressure. Ang2 is going to be produced in the liver, but then it's going to get activated, right, because it gets angiotensinogen, and then it has to get activated in the lungs with our ACE, right? The plasma protein of the liver is going to release it, and again, it's circulating all the time. What's it going to do is that in the proximal convoluted tubule and the ascending loop of Henle, and in the collecting duct, we're going to reabsorb more sodium and therefore more water. Okay? So our overall body response is that we're going to have less water and sodium loss. And so by doing that, we're going to increase our blood volume and therefore increase our blood pressure. Renin is released by the kidneys, and they're going to be released when the JGA cells, those juxtaglomerular apparatus cells, are going to detect low blood pressure. What it's going to do is that it cleaves that angiotensin to Ang2, and we get that artery constriction, so vasoconstriction. It's going to decrease our glomerular filtration rate, and it's also going to increase thirst, which is therefore going to increase our blood volume and hopefully increase our blood pressure. So these top are going to all increase blood pressure. That's the goal. Atrial natriuretic peptide is released at the heart, and it does that in response to stretch. It can sense that increase in blood pressure. And so what it does is it increases our glomerulation filtration rate, which is going to decrease our sodium chloride reabsorption in the distal convoluted tubule, and it's going to inhibit renin. So it turns off the renin here. So more water and sodium loss is going to, if we get rid of it through our urine, that's going to then cause us to have a decrease in blood volume and therefore a decrease in blood pressure. So here, right, is that when we have this atrial natriuretic peptide, it's going to sort of turn off the renin. Okay, so the atrial natriuretic peptide is going to turn off the renin, which is going to increase our glomerular filtration rate. Okay, so atrial natriuretic peptide decreases renin, which can then increase our glomerular filtration rate. Whereas renin overall is going to decrease that glomerular filtration rate. Okay, and so all these green ones, they are going to cause us to hopefully raise the blood pressure, and the red one is going to cause us to lower the blood pressure. Okay, so that's the goal. So what are the structures of our urinary system? The urinary system, we also call it the renal system, and it's going to consist of our kidneys, our ureters, our bladder, and the urethra. And each kidney is going to consist of millions of functional units called nephrons. They're the nephrons, right? And so the nephrons are hanging out in this region here. The urine is going to be created with filtration. So first the small molecules get filtered from the glomerulus into that BOWMAN'S capsule. Then we get reabsorption through those nutrient molecules. They get transported from our proximal convoluted tubule and our distal convoluted tubule to our paratubular capillaries. We are then going to concentrate our urine so the water gets reabsorbed from the descending limb of the loop of Henle, is what it's supposed to say, and the collecting duct into those paratubular capillaries. And then we can do secretion, which is our waste or harmful substances get transported from our paratubular capillaries to our proximal convoluted tubule and our distal convoluted tubule. And then we can do excretion, which is where we actually are going to urinate. Okay. Now, if you have any questions, you know where to find me. I'm happy to help.